The nervous system is divided into the central nervous system, CNS, and the peripheral nervous system, PNS. The CNS is made up of the brain, encephalon, and the spinal cord. Its development begins very early during embryogenesis, starting from one of the three main embryonic layers, the ectoderm. Around the third week of embryonic development, part of the ectoderm thickens to form the neural plate. This plate folds inward along the midline, creating the neural groove, while the edges rise up to form the neural folds. By the fourth week, the edges of the groove gradually join along the dorsal side of the embryo, forming the neural tube, a hollow structure that closes by the end of the fourth week. During the closing process, the internal space of the neural tube is called the neurochole or neural canal, which will later develop into the ventricular system of the brain and the central canal of the spinal cord. Around the fourth and fifth weeks, the anterior part of the neural tube enlarges and divides into three primary brain vesicles: prosencephalon, forebrain; mesencephalon, midbrain; rhobencephalon, hindbrain. These vesicles will give rise to different regions of the brain. The posterior caudal part of the neural tube, which remains narrow and elongated, will become the spinal cord. Starting from the sixth week, the three primary vesicles divide further into five secondary brain vesicles. The prosencephalon becomes the telencephalon, which forms the cerebral hemispheres, and the diencephalon, which includes the thalamus and hypothalamus. The mesencephalon does not divide further and gives rise to structures like the colliculi, superior and inferior, and the tegmentum. The rhobencephalon splits into the metencephalon, which forms the pons and cerebellum, and the myelencephalon, which develops into the medulla oblongata or bulb. By the eighth week of gestation, the main structures of the CNS are already well formed. The structures derived from the midbrain, pons, and medulla oblongata constitute the brainstem, which connects the brain to the spinal cord. The cerebellum, although tightly connected to the brainstem, is considered a separate structure. The brain can be divided into telencephalon, diencephalon, brainstem, and cerebellum. The outermost part of the brain is the cerebral cortex, which is divided into lobes. The CNS is protected by three meninges: dura mater, arachnoid mater, and pia mater. The cerebrospinal fluid, CSF, flows between the arachnoid and pia mater in the subarachnoid space, in the brain's ventricles, and in the spinal canal. It helps protect and nourish the CNS. The spinal cord extends from the foramen magnum at the base of the skull to the first or second lumbar vertebra. It is divided into four regions: cervical, thoracic, lumbar, and sacral. The spinal cord has 31 segments, each giving rise to a pair of spinal nerves, 31 pairs in total. It ends with the conus medullaris, medullary cone, from which extends a thin fibrous structure called the filum terminale, which anchors the spinal cord to the coccyx.